Right. Good afternoon, everybody. Today we are back with another edition of PG Masterclass on a very basic topic, requested very specially by my own postgraduate students at MAMC that they wanted something on bacterial virulence factors and no other than Dr. Reba from Pathogenesis. Good afternoon, everybody. Today we are back with another edition of PG Masterclass on a very all right. So over to you, ma'am.
Yeah. Okay. Thanks, Sonal. And good evening, postgraduate students. I hope you find this discussion on bacterial virulence mechanisms useful. And I must thank Dr. Sonal and the IAMM for initiating this program, which I think is extremely useful and should be, should help all the postgraduates across the country. I think it's a very unique initiative by the association. So I hope all of you will make full use of this. All right, so today we will be discussing on the bacterial virulence mechanism. We will talk about, we'll define what are the virulence factors, or rather, what is a virulence factor. We shall classify some of the bacterial virulence factors. We shall look at toxins, and then we will also look at the importance of these virulence factors in microbial survival, mechanisms of action on human cells at the macro and molecular level, and finally a little bit on the genetic regulation of these virulence factors. Now, before we go into looking at the virulence factors, first we must know what exactly is virulence. Now, all of you, we talk about, so and so the Vibrio cholerae is a very virulent organism, or Salmonella is less virulent. So how do we define this virulence? Virulence actually is a quantitative measure of the pathogenicity. Now, pathogenicity, or rather pathogenesis, is the ability of the microorganism to produce or to bring about changes whereby it produces certain pathological changes within the cells, which is beneficial to the bacteria and harmful to the host. So the virulence, therefore, is a quantitative measure. We talk about virulence. This organism is more virulent than the other. So how do we measure this virulence factor? This is measured by the lethal dose. And what is this lethal dose? The lethal dose is actually the number of organisms which are required to cause a disease. And which in other words, we call it as the infectious dose. Now, this lethal dose is actually measured in animal experiments when we inject a particular toxin or a virulence factor into the animal, a group of animals, and we wait to see what percentage of the animals die. So if 50% of the animals die, we call that the dose of virulence is, the lethal dose is LD50. And if all 100% animals die within a specified time with certain clinical features and pathological features, if all 100% of the animals die, we call it as LD100, which is extremely virulent. But however, in most experiments, the researchers stick to LD50. Now, so this means that number of organisms which are required to kill 50% of the experimental animals. Now, just to give you an example, we've all known that Shigella is more virulent than Salmonella because around 100 strains of Shigella are required to produce the disease in comparison to Salmonella, which requires about 100,000 organisms. So therefore, the infectious dose depends on the virulence factors of the organism. And now we will be looking at what are these virulence factors. So the virulence factors are essential components that enable bacteria to first establish an infection, to overcome host defenses, and to cause disease. So it is a series of actions. First, it has to establish an infection or establish itself in the host. Then it should overcome the host defenses, because that is how the host is protected against the invading organism. So we talk about our immunity. So it has to overcome the host defense mechanism. And then finally, it should be able to establish the disease and cause infection, cause the disease. So this is the sequence of events which happen or which occur when the bacteria, a virulent bacteria, causes an infection. So these are the virulence factors. Now let us look at what these virulence factors are. Now these virulence factors are functionally classified as toxins, immune evasion factors, adhesion factors, and invasion factors. Now, if you look at the above three statements which I have mentioned, establish infection, overcome host defenses, and cause disease. So the immune evasion factors are the ones which overcome the host defenses. And established infection is through adhesions, and cause disease is through invasion factors. So they are all very closely linked, and it is a series of actions which occur, which are based on the microbial structure or the microbe itself. So these are the essential components of the virulence factor. So essentially, array of virulence factors encompass a wide range of molecules produced by the pathogenic microorganism. So therefore, virulence factor is basically the byproducts of the microbe, which is able to establish or cause changes within the host. So therefore, these factors are basically the ability to evade the host defense mechanism. Actually, it has to, protect itself from the host defense mechanisms, from the cell-mediated immunity, from the humoral immunity. It should be able to protect itself. Otherwise, it will get destroyed and will not be able to establish an infection. So whether it's a virulent or an avirulent organism, it will be removed from the body. Then next step is help the bacteria to thrive and flourish in the host. Naturally, the bacteria has to survive in the host to be able to establish an infection and to become virulent. So therefore, the virulence factors are those which help the bacteria, first of all, to evade host defense. Second, to be able to thrive and flourish in the host. And after establishing itself, finally, it leads to the establishment of infection, which can often be fatal if not treated or if not removed controlled. So we now next move on to the virulence factors. Sorry, Dr. Sonal, can you move on to the next slide, please? The virulence factors, the virulence factors include secreted products such as toxins, enzymes, exopolysaccharides, some of which have the ability to enter the host cell and manipulate intracellular machinery, contributing to the success of the infection. Example, the cholera toxin. Now here what happens? These virulence factors are secreted products of the bacteria. They are not structurally part of the bacteria. It's not the bacteria per se which produces the virulence, but it is the bacterial products such as the toxins, the enzymes, the exopolysaccharides. And I give you an example, the cholera toxin. The cholera toxin, the toxin enters the host cell and causes the changes. It's not the Vibrio cholerae which enters into the host cell. Or rather, many of the bacteria, the bacteria do not enter the host cell, but they secrete certain substances which enter the host cell or damage the host cell, thereby establishing the infection. So this is the secreted products which are the virulence factors. And they assist in tissue injury the same way. Proteases, lipases, they destroy the lipid content of the cells, the protein, they denature the proteins, thereby causing the cell damage and death. Next slide. So the cell surface structures, like I mentioned, one is the cell product, the toxins, the lipases, the proteinases. Next, we come to the cell surface structure. What are these structures which act as virulence factors? We all know the capsulated bacteria, capsulated microorganisms can survive within a macrophage and not get destroyed. For example, the pneumococcus. So these capsules, like the lipopolysaccharides, the glycopeptides, the lipoproteins, all of these together act as the virulence factor of some of the bacteria, like the pneumococcus, the capsulated microorganism. So then we next come to the intracellular metabolic regulatory network. Now, these are governed by protein sensors and regulators. So having entered into the host, the microorganism produce an array of virulence factors to interfere with the host defense. So these are basically the intracellular metabolic regulatory networks of the bacteria, which cause virulence, which cause the death of the cells which they invade. And then we have the non-coding regulatory RNAs, which contribute to the virulence. Now, these non-coding RNAs are not basically the structural RNAs of the bacteria, which are responsible for the replication, etc. But these are the non-coding regulatory RNAs, which regulate certain enzymes, certain toxin productions, etc. They contribute to the virulence of the organism. So then, therefore, clinically, the virulence of a pathogen refers to its inherent capacity to provoke specific clinical manifestations that can be linked to the production of a virulence factor. Now, just to give you an example, we know about the toxic shock syndrome toxin of Staphylococcus group A. Now, these are the virulence factors which are specifically target certain cells and produce various physiological changes within the cell, thereby provoking clinical manifestation and leading to disease. So here we have these virulence factors which are very specifically targeted towards certain manifestations of the clinical spectrum. Now, the other virulence factor is the quorum sensing mechanism. Now, quorum sensing mechanism is basically for the bacteria to be able to spread, to be able to invade locally. So, in addition to the secretory factors and the other cellular factors, the bacteria are able to sense the cell-to-cell communication and thereby produce cell-to-cell communication and act as invasive property of these act as the invasive property of the bacteria. So bacterial outer membrane vesicles are recognized as an efficient means of bacteria to bacteria and bacteria to host communication, not only interspecies but also interspecies. So therefore, by communicating, the bacteria are able to attack or invade the neighboring cells of the host, or they are able to form like most common example of this is the biofilm production. The biofilm production, of course, is also a protective mechanism for the bacteria, whereby the host defense mechanism fails to reach the bacteria. So therefore, this also forms part of the virulence mechanism. Next slide. Now, the other, we move on to the next virulence factor, which is the adhesion factor. The adhesion factors are substances that allow bacteria to adhere to the host cells or elements of the extracellular matrix, promoting colonization and infection. So these are pili, fibrae. Pili, we know, especially the uroepithelial cells, the uropathogens, the Escherichia coli, which are uropathogens, they have the pili, which allows the bacteria to adhere to the mucosal membrane and not get washed up by the urine or other liquids in, let's say, in the intestine. So therefore, pili also act as one of the very crucial virulence factors in many of these bacteria which colonize the mucosa. some of the respiratory pathogens also have these pili. And fimbriae, on the other hand, is very crucial again for the bacteria to move away from host defense structures or towards the host epithelial cells or target cells. So both pili and fimbriae, they are adhesion factors, and they contain these specific proteins which act as the virulent factor of that particular organism. And these proteins that make up the fimbriae and pili can differentiate, are different between bacterial species, and therefore they enable various bacteria to bind to various host cells. That means they are host cell specific pili and fimbriae. To give you an example, Pseudomonas aeruginosa, which causes pneumonia. Now, normally we know Pseudomonas aeruginosa, which is an opportunistic pathogen, causes infections in any part of the body, including cellulitis, etc. But it can also produce pneumonia. So those Pseudomonas aeruginosa isolated strains which can produce pneumonia, they do so by attaching to the respiratory epithelial cells by virtue of the pili. So therefore, that makes it a very potent virulent factor of Pseudomonas. Then there are non-protein containing adhesion factors also, like the polysaccharides and the capsules, as we have seen in pneumococcus, and the gram-negative bacteria like Escherichia coli, which has lipidase. So all of these also are non-protein containing adhesion factors, and they act as virulent factors for the organism. Next one. Next slide. Yeah. So then we have, after the adhesions, then we have the invasion. Now, having adhered onto the mucosa or to the host cell membranes of cells, the invasion factors now come into play. These components include the effector proteins that change host cell signaling pathways and allow the bacteria to survive. And they also have these, they have the effector proteins, and then the secretion system, which deliver bacterial proteins into the host cell to affect cellular processes. Now, bacterial proteins, example, some of the toxins, exotoxins, which are protein in nature, they are delivered into the host cell through certain secretion system, and also the effector proteins which change the host cell signaling pathways, allowing the bacteria to survive within the host. So these are the invasion factors. To give you an example, Salmonella enterica, it has a type 3 secretion system which allows it to enter the host cells and release certain effector proteins that alter the host cell signaling pathways to aid in the bacterial replication and survival. So this is a good example of an invasion factor, which is a effector protein like the type 3 secretion system, which allows the bacteria to replicate within the host cell by altering the host signaling pathways, whereby that would have killed the microorganism. Then the other important factor that we talk about in case of virulence factor is the motility of the bacteria. Now they move through host tissues and acquire fresh niche for colonization. The bacteria migrate through liquids also, like the host tissues, in the host tissue by help of flagella. So therefore, moving the movement of the bacteria is also one of the virulence factors. And actively, the more active the bacterial movement, like the Vibrio cholerae, we have seen how rapidly it moves, that the motility of this bacteria also helps in colonization. It is able to hide in certain niche within the host mucosa, thereby evading the host defense mechanism, and also helps it to enter into the bacteria by moving into certain target cells. So another example is gastritis by Helicobacter pylori. It utilizes flagella to pierce the mucous layer of the stomach. So these are some of the examples whereby the invasion factors like the pili, the flagella, and which help in the motility are responsible for the virulence of the microorganism. Next slide. Now, the immune evasion factors. Now, we have seen how the bacteria, from the bacterial end, the virulence factors act. Now the other inhibitory factor for the bacteria to establish an infection are the immune system of the host. We have the innate immunity and the acquired immunity, the humoral immunity, cell-mediated immunity. So various immune mechanisms within the host allow the host to be protected against these invading bacteria. So therefore, for the bacteria to establish an infection, it should be able to evade these immune mechanisms. Right? So the various factors which allow the bacteria to subvert or control the immunological response of the host, they are very important virulence factors. They can prevent complement activation by creating proteins that bind to and inactivate the complement protein. This is a very good example of evading host immune mechanisms. We know that complement activation is one of the very crucial parts of the host immune mechanism, the complement system. And many of these bacteria, they inactivate the complement factors and thereby inactivating the complement system and establishing an infection. So the example of which is a Staphylococcus aureus. It produces a complement inhibitor protein that binds to the C3 component of the complement and prevents its activation. And we know in the complement cascade how the complement cascade moves from one C1 to C2b, then C3, which divides into C3a, C3b, and that goes on. And finally, the bacterial cell gets lysed by formation of micropores. So therefore, what happens is when Staphylococci infect or invade the host, they bind to the C3 component of the complement, and therefore the cascade stops there, and it cannot move on to the classical complement pathway And the next by evading human host mechanism is by producing proteins that prevent phagocytosis, thereby eluding host immunological mechanism. Here, a very good example is the Streptococcus pyogenes M protein that binds to the complement factor H, which is a regulator of the alternate complement pathway. We know that complement has the classical pathway and the alternate pathway, which are very vital for the host immune system. So we have the Streptococcus pyogenes M protein, and that binds to the complement factor H, and thereby it inhibits opsonization and phagocytosis of Streptococcus pyogenes by the host immune cell. So these are the immune evasion factors which are responsible for bacterial invasion to the host and act as a very potent virulence factor. Next. Now, importance of virulence factors in bacterial pathogenesis. So having looked at the virulence factors per se attached to the bacteria, now let's have a look at the pathogenesis that they produce in the host. So impact of virulence factors on host cells and tissue. Type 1 fibria of E. coli increases the bacterial pathogenicity for the urinary system by encouraging adherence to uroepithelial cells. I've told you earlier that we have these uropathogens, the Escherichia coli uropathogens, which have type 1 fibriae, and these type 1 fibriae allow the bacteria or help the bacteria to adhere to the uroepithelial cells, and thereby they do not get flushed out with the flow of the urine. The other important, adhesion factor, virulence factor for adhesion to the host cell is the lipoarabinomannan of Mycobacterium tuberculosis. This arrests the phagosome maturation. Now we know that the mycobacteria are intercellular pathogens. Now, when they enter into the cell, they will naturally get killed by the phagocytes. So therefore, this LAM or the lipoarabinomannan, which is Mycobacterial tuberculosis specific, it arrests the maturation of the phagosome and enables the bacteria to multiply inside the macrophages, preventing host immune response. So these are two important virulence factors in the bacterial pathogenesis. And then next, moving on from pathogenesis to the disease progression. So the role of virulence factors in disease progression, virulence of a pathogen has the capacity to provoke specific clinical manifestations which are linked to the production of virulence factors. So therefore, therefore it goes on right to the end of its pathogenesis in evoking specific clinical manifestations. Like for example, group A strep, which produces cellulitis. Now, when it invades the host, the group is strep that secretes certain toxins, which manifest, which affect the muscles and the subcutaneous tissues, and thereby producing cellulitis. So, when we know that this cellulitis is an outcome of one of the virulence factors of Streptococcus group A. Then toxins like cholera toxin. We have seen the mechanism of cholera toxin and how it produces diarrhea, the effluent, the efflux mechanism, and the outpouring of sodium and potassium and fluid loss, etc. And that is caused by a specific cholera toxin. So the outcome or the clinical feature that we see in cholera is due to the virulence factor of cholera toxin. Next. For this, we've already been mentioned, but just to highlight, quorum sensing is communication system, and it is known to regulate number of bacteria such as biofilm formation, motility, virulence. Now, when you move on to the next slide, please. The genetic role of the genetic control of virulence factors at the molecular level. Now, we have looked at the virulence factors which are responsible, which are microorganism specific. It's part of their structure, like the capsule, the fimbriae, the phyla, etc. So these are general category of virulence factors, and their control is through the genes which are encoded in the bacteria of various species. Like for example, Vibrio cholerae codes for the toxins, cholera toxins, the fimbriae, the type 1 fimbriae coded for by in the Escherichia coli, uropathogenic E. coli. So these are all genetically regulated virulence factors found in pathogenic strains of species and not in non-pathogenic species. So therefore, this helps us to differentiate a pathogenic species from a non-pathogenic species based on looking at the genes. So therefore, detection of virulence factors can does depend on detecting the virulent gene in the bacteria, like the tox gene in Vibrio cholerae that tells us that this particular isolate is a toxic isolate or a virulent type. Now, the other way of looking at it is inactivation of the encoding gene in the animal model can attenuate the virulence of the strain. So therefore, we now have looked at the genetically modified vaccines. Now, what are these genetically modified vaccines? The genetically modified vaccines are based on the attenuation of virulence by modifying the genes which are responsible for the virulence factors. So inactivating these genes will cause the bacteria to become avirulent. And the next step is when we reintroduce or reactivate these genes, the bacteria becomes virulent again. So that is when we see these epidemic strains of certain organisms which have turned avirulent over a period of time, suddenly become virulent because of the reactivation or reintroduction of the virulent genes into their system, into their cells. Now, some other factors which are responsible for virulence and are also not found in the avirulent organisms. So, human commensal bacteria, as an example, they do not possess virulence factors under normal conditions, like the Coagulus negative staph, which colonize our skin, or the bacteria, the oral flora. So they are normal commensals. So therefore, they do not have the genes coding for the virulent factors. Whereas under certain conditions, under specific situations, they can become virulent. And that is, say, for example, by cutaneous barrier breach. You have, when there is a cut or a breach, the coagulase negative staph can enter and they can become virulent. And here the virulence factor is mainly because of the lowered immune status of the host, and not because they start generating certain newer, toxins or enzymes. And the staphylococci are examples of such dual characterization of virulence factors. Next slide. Now we so the bacterial toxins. So having looked at the structural elements of virulence factors, now let us look at the byproducts of the bacteria, which act as toxins. So bacterial toxins are elements that inflict pathogenic characteristics to some microbes. They are genetically controlled. The capability of the microbes to produce toxins is called toxigenicity of the organism, and they cause adverse effects in a variety of diseases. And we know it's a basic undergraduate level information that toxins are exotoxins and endotoxins, which have varied type of mechanism of action. Generally, the endotoxins across the spectrum of bacteria, the outcome or the action is uniform throughout, whereas the exotoxins have specific targeted mechanisms of action and differ from bacteria to bacteria and the cells that they affect. Next slide. Bacteria release exotoxins into the extracellular environment, and these exotoxins can be further divided into groups according to how they work. Like, for example, the cytotoxins. Now, the cytotoxins cause host cells to die by rupturing or impairing their metabolic system. The cholera toxin produced by Vibrio cholerae is an illustration of a cytotoxin. It results in diarrhea by interfering with the ion transport of the gut. So cytotoxins, there are several bacteria which have, which act on the cells directly by causing toxic effects to the cell by changing their internal milieu, like the ions and the cations and the anions. They change. The bacteria therefore becomes, osmotically unstable. I mean, the cells, not bacteria, sorry. The cells become osmotically unstable. They exude the intracellular fluid, and causing that results in the cell death. And clinically, it is in the form of diarrhea. Now, the other toxin is superantigens that damage the membranes. Now, superantigen toxins can cause systemic inflammation and shock by activating an overactive immune response. That's why it is known as superantigen. I'll give you an example subsequently how the superantigens act and what are the organisms containing the superantigens. So then we have the membrane damaging toxins like the hemolysins, which destroy the red blood cells. We have the E. coli, Escherichia coli producing hemolytic toxins, and the streptococci producing hemolysis, which are toxic. So a large number of microorganisms have these membrane damaging toxins in the form of hemolysis. And then mucosidins, which destroy the white blood cells. Now, bacterial protein toxins activated at the cell membrane. Now, transducing a transmembrane signal and hormone-like toxin. This is an example of bacterial toxin acting as a hormone, or it acts as hormone-like and is thermostable enterotoxin type A of the endotoxigenic E. coli, which is responsible for diarrhea in children and adults. So, transducing a transmembrane signal and acting like a hormone is this E. coli STA. Now, the membrane damaging toxins, bacterial membrane damaging toxins encompass pore-forming toxins. And enzymatically, they activate the toxins against membrane compounds. They are the main toxins of bacteria involved in gangrene, myonecrosis, and soft tissue infection. So these toxins actually bind to the cell membrane and produce pores within the cell, on the cell's cell membrane, and thereby they cause cell death, myonecrosis, and soft tissue infection. Certain pore-forming toxins interact specifically with intestinal or lung epithelial barrier, leading to specific diseases, like some of these respiratory pathogens. They have these pore-forming toxins which attach onto the alveoli and cause exudation of the fluid and leading to inflammation and infection and inflammation. Now, next one. Yeah. So now coming back to going a little to the detail of the superantigens, which are very, very potent virulence factors of some of the bacteria, like the streptococcus, the staphylococcus. The superantigens are a family of potent mitogenic toxins. They are 20 to 30 kilodaltons. This is the textbook information. They're all powerful T-cell mitogens. They activate the immune system by binding to the major histocompatibility complex class 2 and the T-cell receptor molecule. So as you can see here in the diagram, sorry, I don't have a pointer. I can't because it's with the so we have the conventional T-cell activation by the antigen, by the microorganism contained within the antigen-presenting cell, which stimulates the T cell, and thereby it is a conventional method of cell activation where only 0.01% of the T cells are activated. Whereas on the other hand, you have the superantigens, which are T cell mediated again, but here what happens, these superantigens have the property to attach to the major histocompatibility class 2 antigen, and thereby they stimulate larger number of T cells, and therefore larger number of T cells are activated. Almost more than 20% of the T cells within that zone are activated by the superantigens. And you know the T cell alpha and beta, and what are the activities? They are cytokines, chemokines, inflammation, fever, and they also give rise to scarlet fever, B-cell apoptosis, decreased immunoglobulin synthesis. So these are some of the adverse outcomes of the superantigen stimulate of streptococci, which are responsible for the virulence of that particular group of organisms seen in group A strep. This staphylococcus superantigens also are very, very potent. They are responsible for staphylococci endotoxins, which produce food poisoning and toxic shock syndrome. And they also act in a similar way, whereby they act with a type 2 major histocompatibility antigen and thereby secrete various interferons, interleukins, and alpha, beta, stimulate the T cells to produce all these molecular mediators or cytokines to cause infection. And as you can see here in this diagram, also a normal antigen presenting cell with a normal organism, invading organism, which does not have superantigen, stimulates about 0.01 to 0.1% of cells which are activated. On the other hand, the superantigen attached to certain as a virulence factor of certain microorganisms, and in this case, the Staphylococcus aureus, you find that 20 to 30% of the T cell population are activated, and thereby there will be a concomitant increase of the interleukins, interferons, and killer cells, etc., which are stimulated to produce during this particular action, and therefore they lead to some of the very lethal infections like toxic shock syndrome and food poisoning. Next slide. The other form of exotoxins are the bacterial pore-forming toxins. Pore formation through cell membrane is the most common mechanism of bacterial toxins. About 30% of bacterial toxins are pore-forming toxins. They have two main classes, alpha pore-forming and beta, according to their structure-based interaction with the lipid layer of the cells. Now, these pore-formings are secreted as soluble monomer. Then they bind to specific receptors on the cell membrane. They oligomerize and then form the transmembrane pores. And these pores are extremely potent toxic outcome of a bacterial invasion, and they allow the cells intracellular material to exude out from this from the cells and leading to cell death. So Clostridia, Staphylococci, they all have pore-forming toxins, and this diagram is an example of a pore-forming toxin. Then the other toxin that we all know about are the neurotoxins, the clostridial neurotoxins, which impair the neurotransmitter release. And they specifically target neuronal cells and impair the release of neurotransmitters, like the botulinum neurotoxins or the tetanus neurotoxins. And as this particular diagram shows, that the toxins migrate, they can transfer, they can sort of migrate from the neurons through the central nervous system, causing central inhibitory in manifestation. So you have these specifically targeting toxin virulence factors in the form of exotoxins. Now, finally, the toxins which induce cell death. Now, cell killing is a major effect of bacterial toxins. Various strategies are used by bacterial toxins to induce cell death. Like, for example, diphtheria toxin. We know that diphtheria toxin causes protein, prevents protein synthesis in cells and thereby leading to cell death. So, the diphtheria toxin is one of the most potent cytolethal toxins. The Pseudomonas aeruginosa exotoxin A shares a similar mechanism of action with diphtheria toxin, despite a different structural organization. So now you see the toxin, even though it is secreted by different classes of microorganisms, but their mode of action and their structural resemblance exists between unrelated bacteria. The Shiga toxin is produced by Shigella dysentery, and a similar Shigella-like toxin produced by enterohemorrhagic E. coli, such as the E. coli 0157. So toxins are very, very potent virulence factors, irrespective of the organism from which they are secreted from or formed by. Next slide. So now, just an example of the pathophysiology of exotoxins of Escherichia coli, because these are some of the very important, toxins, and I think it's a favorite question among the examiners regarding the toxins of Escherichia coli. So there are 7 pathophysiotypes of toxins of exotoxins of Escherichia coli, like the enteropathogenic, enterohemorrhagic, enterotoxigenic, enteroinvasive, enteroaggregative, diffusely adherent Escherichia coli, and a new pathotype, which is the adherent invasive E. coli, AIEC, that is an adherent invasive E. coli, which is a new pathophagiotype. They cause mostly diarrhea and intestinal disorders, but also it is not basically localized to the intestinal, because as we know, the enterohemorrhagic E. coli pathotype has also been implicated in extraintestinal disease like hemolytic uremic syndrome. So these are some of the clinical manifestations of a similar type of toxin, like the enterohemorrhagic E. coli. It produces diarrhea as well as hemolytic uremic syndrome. So a same toxin having different clinical outcomes. Now, so finally we come to, or not finally rather, the other important toxin, endotoxin. Like as I had told you earlier, although endotoxins are secreted by various types of bacteria, but the outcome and the mode of action as well as the mechanism are similar across the board. So these endotoxins are basically inherent part of the bacteria. So the bacteria needs to be present at the site of infection for the endotoxin to be active. So they form the component of the outer membrane of gram-negative bacteria, mainly seen in gram-negative bacteria. They are released into the circulation upon disruption of the intact bacteria. Now, this is very crucial. Like those toxins, those bacteria which are found within the normal, normal physiology of the bacteria, normal, body parts. When the bacteria die, they can produce endotoxin. So these toxins are released into the circulation upon disruption of intact bacteria. Endotoxins are especially abundant in the gut, naturally, because our gut is colonized by myriads of gram-negative bacteria, and therefore they are abundantly found in the gut, the tissue source of small intestinal submucosa. And not only, the gut, specifically they form the tissue source for small intestinal mucosa. Endotoxin is not reliably destroyed by, this is very important, that they cannot be destroyed by disinfection, steam sterilization processes, or ethylene oxide sterilization. This again becomes an important issue in hospital infection control or hospital-acquired infections where, endotoxic, like for example, in the IV fluids, in the other intravenous fluids, endotoxins can be present even though the material has been sterilized, and therefore they can produce a very serious damage, serious outcome in the host. So endotoxins, however, can be destroyed by plasma exposure to inactivated endotoxin. Of course, their work is going on to show that they are promising the plasma sterilization. And what do they cause in the host? They cause inflammatory reaction in the host. So what happens? The macrophages encounter the lipopolysaccharide molecules or the endotoxins, and then they release cytokines that stimulate inflammation and draw other immune cells to the infection site. So this is basically the mechanism of the endotoxin in general. So again, graphically, the endotoxin, especially the lipid-A, it activates the macrophages, like as I had mentioned, and these macrophages therefore release interleukin, which causes fever, tumor necrosis factor, which causes fever and hypotension, and nitric oxide, which gives rise to hypertension. So therefore, we see when there is endotoxemia, the patient generally has fever, and hypotension is again another very characteristic feature of endotoxemia. Then endotoxins also activate the complements, and like the C3A produces hypertension, edema, and C5A, which produces neutrophil, which causes chemotaxis of the neutrophil. And also it activates tissue factors like the coagulation cascade, and therefore that leads to the disseminated intravascular coagulation, which we normally see DIC catastrophe in endotoxemia in septic shock caused by gram-negative bacteria. So this is basically, I have taken this from the Javits clinical microbiology book, and all of you can, so here basically I would, you can look up all these materials which I have produced here, or rather reproduced. I'll give you the reference at the end, but there are some textbooks which will help you to go through and read in detail and understand the mechanisms of these various virulence factors. Now, finally, we come to the regulation of virulence factors. The expression of virulence factors is regulated by genetic and environmental. It's not just only genetic. As we have seen, if the host environment is conducive, the bacteria becomes virulent. So therefore, the genetic regulation of virulence factors is regulated by transcriptional factors that respond to environmental signals, like for example the FIM operon, and for production of type 1 fibrillary in the pathogenic E. coli. So when there is an environmental signal for the bacteria to produce pathogenesis or to become pathogenic, that is regulated by the transcriptional factors. And an example is the phenopyrrole, which gets activated to regulate the type 1 fibrillation. What are the environmental factors that influence virulence expression? It is a temperature, pH, nutrition, availability of nutrition rather, and oxygen levels. Example, low pH and high temperature promotes the production of cholera toxin in Vibrio cholerae, mimicking the conditions encountered in the human small intestine. So these are the environmental factors that can influence virulence expression in the pathogenic bacteria. Now, before I close, there is something like I had, if you recall, I had told right in the beginning, the epidemic pathogens. Now, what are these epidemic pathogens and what are these epidemic, the virulence factors of these epidemic pathogens? Epidemic bacteria are highly specialized species that are adapted to their host and are characterized by extensive genome alteration. That is, they lie like, they are kind of dormant, and so that they are able to survive within the host or in the environment without being, recognized or without being removed or destroyed because of their virulence. Now, they possess a damaged recombinant system, but significantly, they are more toxic and more toxic. Rather, they produce more toxin. So they produce a recombination factor and a repair system, so that when stimulated, they're able to produce more of the virulence factors, example, the more of toxins. So they mostly lose the genes related to metabolic activity, the production of energy, cell motility, and transcription than the closely related non-pathogenic or non-epidemic. Now, this just as I had told you earlier, they remain rather very dormant, almost as if they're hiding without disturbing the outside environment, so that they are not noticed. But when the environment becomes conducive for them, then their genetic mechanism acts up and they produce more toxins, more virulence factors than the avirulent or the non-pathogenic or non-epidemic species, which are found in non-epidemic forms or in non-epidemic periods. So, with that, I think I've just covered mainly only the virulence factors. I haven't covered how to detect these virulence factors and what are the various laboratory techniques to detect that, because that again is a huge topic and evolving almost every day. And these are some of the references which I have looked up to prepare this particular presentation. But I would request all of you to read up the book that is the Review of Medical Microbiology and Immunology by Melnick and Warren Levinson. These are two books, and I found the virulence mechanisms and the explanations are very lucid, and it will help you to form the basis of your preparation for your examination plus your knowledge also. So this is a very good book at the postgraduate level to read up the basic virulence mechanism. So if there are any questions, next slide, of course, goes into any questions. I'm sure we are online, so therefore it's not one-to-one, but I've given my email address at the top. If any of you want to get in touch, you are most welcome. And it's really been a pleasure. I wish it was a offline class where I would have interacted with all of you. But anyway, thank you, Dr. Sonal, for this wonderful expose to the postgraduates.
Ma'am, thank you for making such a difficult topic so simple. And I will now request Dr. Bharti to give the closing remarks. I think ma'am has joined. Any questions you can type in the chat box. I think we all have. Ma'am, there is one question which is more lethal: plasmid mediated virulence factors or chromosomal virulence factors?
Pardon?
Somebody is asked which is more lethal: plasmid mediated virulence factor or chromosomal mediated virulence factor?
Actually, plasmid mediated virulence factors are basically in response to the changing environment. So therefore, it can adapt itself to the changing environment, because plasmid is a transmissible gene, whereas chromosomal factor is inherent to the bacteria. So therefore, it is more like it is inherent to the bacteria. So whichever force the bacteria is attacking or is causing an infection, it will react only to that. Whereas plasmid can be modulated. It can be transmitted from one bacteria. The virulence factor can be transmitted from one bacterium to the other. So therefore, I would say that plasmid-mediated virulence factor would be more virulent, because the ability for the virulence to spread is more.
Yes, thank you, ma'am. Any other queries? You can put it in the chat box. I'm just waiting for Dr. Bharti to unmute. Ma'am, we can see you, but we can't hear you.
Hello.
Anji ma'am, now
Can
we
you
can.
hear me? Yes. I was just trying to figure out my icons. Oh, thank you so much, ma'am. Reba, ma'am, such an excellent talk it was. Really enlightening and really up to date till the last, and everything was so very well explained. So we were really honored to have you, ma'am, in the master class, and thank you for the time taken out for us. Thank you so much, ma'am.
It is my pleasure. Thank you very much for considering me to interact with the postgraduates. And thank you very much, Bharti and Dr. Sonal. It is my pleasure. Thank you.
I think it's a great privilege to have you, ma'am. And I think it's a great initiative, Sonal, that we can really, listen to the experts, and the students and the young faculty can really, and we oldies can really benefit from because so many things which we really miss out, and that so we can really listen to the experts and the ligands directly. So that's a great thing. Great initiative, I must say.
In the chat box that they would like a part 2 of this session. So please also write what would you like, and then we can request ma'am to take a part 2 of this masterclass.
Yes, we would love to have that. We're so honored to have you with us, and it's a great privilege to listen to you. So,
my pleasure. Of course, I'll be happy to have continue this anytime. Since I'm retired, I have a lot of time on hand. I mean, not
keep you busy.
I'm still doing some voluntary work also. But yeah, I would be happy to take on any other session that you feel you need to.
Great, ma'am. Thank you so much, ma'am.
Thank you, Sonal. Thank you, Dr. Bharti.
Thank you.
Thank you, ma'am.
Congratulations
so much.
on this very good initiative.
Really great. Thank you, ma'am.
Okay, so with that we log off. Okay, Dr. Farah has written what they want: the genes mutations conferring AMR in bacteria. Let's stop AMR for some time. There are other topics in the exam which you have to cover.
Yeah.
So, but anyway, we'll keep this in mind and definitely try and cover in the coming few master classes. All right, ma'am. With this, I'll end.
Okay.
Thanks to.
Thank you.
Thank you. Thank you so